Bismillahirrahmanirrahim. Class, today we will discuss the development of female gametophyte and Archegonium in Ephedra. In last lecture, at the end, we discussed the formation of Megaspore in the nucellus. The female gametophyte is developed from the Megaspore. The nucleus of the Megaspore divides by free nuclear division, and before dividing into free nuclear division, after first division, the nucleus of the functional megaspore divides and two nuclei are produced, and these move towards the opposite poles, and in the center vacuole is formed. Then these polar nuclei, these divide to produce 16 nuclei, and these arrange themselves at the periphery of the central vacuole. These are arranged around on the outer side of the central vacuole. The nuclear division continues, and the central vacuole, it starts to disappear, because in the center the cytoplasm starts to grow. Ultimately the vacuole disappears and the nuclei are distributed evenly in the enlarged megaspore. Means after disappearing the nucleus, all the nuclei, they are distributed equally, evenly in the whole megaspore, which is enlarged now. And then in these nuclei, wall formation is started, which is centripetal. Wall formation starts from the outer nuclei and then it continues towards the center of the gametophyte, and in this way the whole gametophyte, it becomes cellular. In this gametophyte, the cells which are present towards the calyzal end, these are dense and are filled with stored food, and these act as the nutritive tissue like endosperm. And those cells which are present towards the micropylar end, these are watery and vacuolated, and in this region, micropylar zone, the Archigonia are developed. In this diagram, these are the four haploid megaspores which we discussed later, which we discussed in the last lecture. Three become degenerated, and the lowermost, it becomes the functional megaspore. In this functional megaspore, the nucleus divides and two nuclei moves towards the pole, and they again divide, and in the center a vacuole starts to develop. Then these nuclei which are present towards the pole, they divide again and again, and 16 nuclei are produced, and these 16 nuclei, these are arranged on the periphery, on the outer side of the central vacuole, and this is the 16 nuclear stage female gametophyte. Then in the next stage, in the center of the nucleus, center of the female gametophyte, where vacuole is present, cytoplasm starts to grow, and this vacuole starts to disappear, and the nuclei also continues to divide, these divide continuously and many nuclei are produced as a result of this division in these nuclei. This is the stage in which the nuclear division has been shown, and in the center the vacuole is still present, but it starts to disappear due to the growth of the cytoplasm. And when the central vacuole it disappears after production of the cytoplasm in the center, then the nuclei which are produced in large number, they are divided into the whole part of the female gametophyte. They are equally distributed in all the parts of the female gametophyte. After this even distribution of these nuclei, then wall formation around these nuclei starts, and this wall formation occurs around the nuclei which are present on the peripheral side, on the outer side, and then this wall formation it continues around the nuclei towards the center. And in this way the whole gametophyte at the end, when the wall formation is completed around all the nuclei, it becomes cellular. The whole gametophyte it becomes cellular. This is the end of the gametophyte region, which is present towards the micropylar end, and the cells of this region they are generally vacuolated and thin, while the cells which are present towards the classal end these are dense and they contain the stored food, and this region acts as a nutritive tissue. And in the upper part which is present towards the micropylar region, here the Archigonia are developed. In this region which is towards the micropylar end, Archigonia are developed. Now we will see the diagram again in which the development of gametophyte is shown. This is the sporogenous cell in the nucellus, which after reduction division which will produce the tetrad of 4 megaspores. Out of these 4 megaspores, 3 will be disintegrated and 1 will be functional megaspore. And in this functional megaspore, the nucleus will divide and this nuclei will move towards the pole. And in the center, vacuole starts to develop. Here we can see again the nuclear division in the nuclei present in the poles and the central vacuole. Here the central vacuole is more clear and the nuclei are arranged around the periphery of this vacuole, and this is the 16 cell stage where the nuclei are arranged on the periphery and the central vacuole is most clear. And this central vacuole starts to disappear by the formation of the by the growth of the cytoplasm in this part, and the nuclei then which are dividing continuously, they are freely dividing and many nuclei are produced. And after the disappearance of this vacuole, all these nuclei they are distributed evenly in whole gametophyte, and then around these nuclei wall formation starts from outer side towards the central part, and in this way at the end the whole gametophyte it becomes cellular. This part acts as the nutritive tissue and which is towards the classal end, and the part of the female gametophyte which is towards the micropylar end, here the archegonia will be developed. Now this gametophyte, female gametophyte which becomes cellular, the position of this gametophyte is like this. This is the ovule, the nucellus of the ovule, and in this nucellus here the megaspore mother cell is produced which will produce the megaspore, and in the megaspores, from this megaspore, female gametophyte is developed, which is present inside the nucellus, which is the cellular tissue at the end in which Archigonia will be developed. Now we will see the structure and development of Archigonium. In Ephedra, the number of Archigonia, these range from 1 to 3, means 1 to 3 Archigonia are developed in different species. Archigonium is produced from a superficial cell at the micropylar end of the female gametophyte. This archigonial initial, it divides by periclinal division to produce two cells. The outer cell is called the outer primary neck cell, and the lower cell is called the inner central cell. The primary neck cell, it undergoes repeated periclinal and anticlinal divisions, and as a result of these divisions, 30 to 40 cells are produced, which form the neck region of the Archigonium. And these 30 to 40 cells, these are arranged in 4 or more tiers. Now the central cell, which is present below this neck region, it enlarges and its nucleus divides once to form two nuclei. Upper one is the ventral canal cell, and the lower one is the egg nucleus. Ventral canal nucleus and egg nucleus are produced from the central cell, and these nuclei are rarely separated by the walls, means wall formation rarely occurs around these nuclei. And the gametophyte cells which surround the central cell, these develop, divide and produce the two-layered jacket of the Archigonium. This is the archigonial initial. And this archigonial initial cell, it divides by periclinal division to produce two cells, neck initial cell, primary neck cell, and lower one is the central cell. Then this neck cell, it divides by periclinal and anticlinal divisions to produce the 30 to 40 cells, and these cells of the neck region, these are arranged in 4 or more tiers. And the central cell, it divides to produce the two nuclei, ventral canal nucleus and the egg nucleus. Then the cells present around this central cell, these form the two-layered jacket of the Archigonia, and the cells of this jacket, these are thin-walled cells. This is the neck region of Archigonium, and this is the part of the Archigonium where the egg nucleus and the ventral canal nucleus is present. This is also the diagram which is showing the nucellus, and inside this nucellus the female gametophyte is produced, and at the upper end of this female gametophyte towards the micropylar end, Archigonia are produced. Here two long structures are produced, which are the Archigonia. Archigonia are the longest Archigonia which are produced in the gymnosperms. And the cells of the jacket layer, which are thin, these may be binucleate cells. So students, in today's lecture we have discussed the structure and development of the female gametophyte, which is produced from the megaspore cell, and the development of archegonium, which is produced inside the female gametophyte. The structure of archigonia, as we have discussed, it consists of large neck region and the lower part in which the one ventral canal nucleus is present and the other egg nucleus is present, and wall jacket layer, two-layered jacket is formed around this archigonium, and archigonia become elongated structure. Here again we can see the diagram of the female gametophyte and the archigonia. Archigonia, this is the female gametophyte, the lower part of which act as the nutritive tissue like endosperm, and in the upper region long archigonia are produced, and the structure of each archigonium is, it consists of large neck region consisting of 30 to 40 cells, and in the lower part of the archigonium ventral nucleus, ventral canal nucleus is present, and the egg nucleus is present. In the next lecture, inshallah, we will discuss the process of fertilization, pollination, and the fertilization.